we didn't have to face them.
So welcome everybody to the Early Career Award talk, CSEE Early Career Award talk. This is the first of 2 talks that will be going on, one right now, one next week at the same time, same place. I'm really excited to have Dr. Diana Rennison here from the University of California, San Diego. She's the winner of one of the 2 Early Career Awards, and she does some amazing research, and we'll get to hear about it now as she goes through her talk. I'll just say a quick reminder, some housekeeping, that you can feel free to type in any questions you have throughout the talk or at the end of the talk into the chat box below the stream, and I will be able to relay those questions to Dr. Rennison and at the end of the talk. And so we'll have a live question period at the end as well. On that note, I'm going to hand it over to Diana and let her introduce herself and her talk.
All right, thanks so much. So yeah, I'm super excited to be here and given this opportunity. I pre-recorded the talk due to nerves and fear of technical issues. So I'm going to play that for you now, and then I'll be here to answer all your questions. So thanks.
So thanks so much to CSEE for giving me this fantastic award and the opportunity to talk to you all today. So my work is focused on the goal of understanding the core mechanisms that shape the fantastic diversity of life we see on Earth today. To uncover these mechanisms, my work generally utilizes the methods of evolutionary genetics. The goal of modern evolutionary genetics is to make connections between phenotype, genotype, and both to fitness. And there's a number of methods that are used to make these connections. For example, between phenotype and genotype, QTL mapping, genome-wide association, and more recently, transgenics have been used. Commonly, genome scans using metrics like FST or empirical estimates of selection are used to make connections between genotype and fitness. However, hopefully by the end of my talk, you'll believe that empirical estimates of selection are really useful for this. And empirical estimates of selection are often used to connect phenotypes to fitness. Now, it's often really difficult to make all of these connections in any single system. However, I still want to go beyond this and overlay a couple other additional questions to bring this framework a little bit further. So for example, I'm interested in why particular genes are used time and time again over the course of adaptation, and what particular ecological factors are important for generating fitness differentials. So in line with this, there's a couple questions really central to my research program. The first is, how do sources of selection interact to shape evolution and the generation of biodiversity? And why do organisms follow certain evolutionary trajectories? So the talk will be split into 2 parts. Each part will address one of these 2 questions. So part A will look at sources of selection and how they interact to shape the generation of biodiversity. And I'll tell you about an experiment I conducted during my PhD that was recently published, where we investigated the contribution of predation to diversification. And I became interested in predation because I went to the literature and I found all these really great examples of how resource competition had been important to diversification. So we have a variety of different systems in which they've become model systems for understanding resource competition and how it spurs diversification. However, when I looked at all these different kind of textbook examples of adaptive radiation, it was difficult for me to imagine that predation alone was responsible for this fantastic diversification in each of these systems. So I started to consider other species interactions and how they could contribute to diversification. So, for example, predators, pathogens, or mutualists, and parasites could all spur diversification, promoting differences among species in a variety of different traits. And I've worked on all of these factors. However, today, the work I'll tell you about is focused on the role of predators. So the system in which I'm examining the contribution of predation to diversification is benthic and limnetic three-spine stickleback. And these species have originated in a handful of lakes in British Columbia, Canada, and they're a really powerful system to address questions related to diversification and parallel evolution. So as you can see in these figures, we have three different lakes that have benthic and limnetic species pairs. This is the benthic, 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 limnetic, limnetic, limnetic. And in all cases, the benthics across lakes look more similar to one another than they do to their sympatric limnetic ecotype. And we think that this is a product of parallel natural selection. And we know that this divergence has occurred really rapidly. So all of these lakes were colonized by marine ancestors within the last 10 to 12,000 years. And we know quite a lot about the ecology of this species. So we know that resource competition is likely important in the generation of these benthic limnetic pairs, as there's been divergence between the species in a number of different trophic traits. And there's also differences in their diet preferences and habitat use. So the limnetics are found out in the open water portion of the lake where they feed upon zooplankton, whereas the benthics are found in the nearshore environment where they tend to feed on insect larvae and mollusks. And the morphology matches these differences in diet. And we think that these differences in diet and habitat use have actually led to changes in other aspects of the ecology. So we think that these species interact with different species of predators due to their differences in habitat occupation. So limnetics out in the open water, we think most often interact with cutthroat trout, so a vertebrate predator, whereas the benthics in the nearshore environment, in a really highly often vegetated region, we think interact most often with insect larvae such as dragonfly or back swimmers. And there's a lot of differences in how these species would interact with these really different predators. And I'll speak to that in a moment. So another reason why we think predation could be involved in this case is that there's an interesting pattern of morphological divergence. So limnetic individuals have really robust bony armor. So they have long first and second dorsal spines relative to their body size. They have a really long pelvic spine and pelvic girdle, and they tend to have a handful of lateral plates along the flank. Whereas benthics have reduced bony armor, so many individuals completely lack the pelvic spine and pelvic girdle. They often have 1 or 0 lateral plates. Many individuals lack this first dorsal spine, and the spines that they do have are much smaller relative to their body size than what we see in the limnetics. And we think this is related to the types of predators that they encounter. So cutthroat trout are a gape-limited predator, which means they can only eat things they can fit in their mouth. And we think that these spines actually increase the effective diameter of the stickleback, making it more difficult for them to be consumed by this gape-limited predator. We think the lateral plates may protect against any lacerations or compression of the internal organs from this predator, whereas the benthics, since they're not preyed upon, we think, by these cutthroat trout very often, and they're encountering these invertebrate predators, it's been hypothesized that these invertebrates actually use these spines as handholds to grab onto the stickleback. So having these features can actually be a liability when encountering these invertebrate predators. So we think that this has led to the divergence of these traits between these species. So for the purpose of my study, I chose to focus on 2 traits, first dorsal spine and pelvic spine. And the reason for that was that they're the most divergent between benthics and limnetics, and also because we had some candidate genes for these loci. So the specific aim of this project was to determine whether a species interaction other than resource competition could also drive divergent evolution. And we wanted to estimate predation-based natural selection on these 2 phenotypes as well as their underlying genes. So we used a variety of different methods. We used QTL mapping to be able to associate bony spines to their candidate genes. And then we used time series data to look at allele frequency changes and trait mean changes to estimate selection empirically. And we wanted to identify what source selection was, so we manipulated trout predation. So I conducted a manipulative experiment at the Experimental Ponds facility at the University of British Columbia campus in Vancouver. And this facility allowed me to conduct a multi-year selection experiment under fairly natural conditions. So I used 8 pond replicates. These are the ponds here. This is an aerial view. As you can see, the ponds are actually quite large, so they're 25 meters in length. And they contain both an open water and a shallow water zone, so kind of trying to recapitulate a natural lake environment. They also contain many of the natural resources that you would find in a nearby lake that would contain stickleback. They're vegetated. And what was really important is they don't naturally contain any fish, so we were able to manipulate the fish environment. However, there's naturally avian predation and insect predation that we did not manipulate, so all ponds were subject to that. Because the ponds were empty, we were able to create an experimental population of stickleback to introduce. And so I bred pure wild benthic mothers to limnetic fathers to create an experimental F1 cross. And these fish were intermediate in their phenotype. And I did this 4 times to create 4 different families. We have 4 F1 families, and this consisted of hundreds of individuals per family. And I split these families in pairs of ponds. So each family went into 2 ponds. And then these F1 individuals were allowed to breed naturally, so find their own partners and create F2 hybrids. And these F2s were really key to the experiment because F2s are highly variable, with some individuals being very benthic, some very limnetic, and everything in between with all sorts of different trait combinations. And this was important because it created a lot of genetic variation for selection to act upon. So with these F2s in the pond, we did an initial sample, our first measurement, where we collected 50 individuals per pond, and this was our pre-selection estimate. We then introduced trout into one of the ponds within each pair. So we had 8 ponds, 4 with trout, 4 without, so our control ponds. And then we allowed those F2s to breed after 1 year, and they created an F3 generation, and this was our post-selection collection. So we took a final measurement after 1 generation of differential selection with 50 individuals per pond. And then all of the individuals were genotyped and phenotyped. So our prediction from the experiment was that in the absence of trout, so in our control ponds, predation would be primarily invertebrate and armor would be selected against, which is the benthic phenotype. And in the presence of trout, armor would be favored, which would create the limnetic phenotype. And so we should see divergence in evolution between the treatments. All right, so jumping into the results. So on the y-axis here, we have the treatment effect on the evolutionary response. So this is the difference within a family between the control and the trout treatment ponds. And so here, a positive value indicates that the limnetic phenotype of more armor was favored in the trout pond. So that would match our prediction. And a negative value would be the benthic phenotype was favored in the trout pond. So that's the opposite of our prediction. And we have our 2 focal traits. So we have first dorsal spine here and pelvic spine. And so what we see is that for all 4 ponds, pairs, in the trout ponds, longer first dorsal spines were favored more heavily than in the control ponds. So this matched our prediction. So we saw divergence towards the limnetic phenotype. Whereas for the pelvic spines, we saw a heterogeneous response. So in 2 ponds, the limnetic phenotype was favored, and in 2 ponds, the benthic phenotype was favored. So we're not really able to say whether or not trout is important for divergent selection on the pelvic spines. But we are able to say that there's strong divergent selection on the first dorsal spine. So then we want to look at what are the candidate genes underlying this difference, and can we find any evidence of selection on it due to trout predation. So as I mentioned, we did QTL mapping to do this, and we were able to identify 2 peaks in the genome, 1 for each trait, that corresponded to some previously identified candidate genes. So on chromosome 4, for first dorsal spine, we identified MSX2A, and on chromosome 7, for pelvic spine, we identified PIDX1, which had a really, really large effect on the variants. So we decided to look for selection at these 2 candidate loci by looking for changes in allele frequency between the F2 and the F3 generation. And what we found was that for MSX2A, our first dorsal spine candidate gene, in all 4 trout ponds, the limnetic allele was more favored than in the control ponds. So we had positive selection for the limnetic genotype. And for the pelvic spine candidate at PIDX1, we found a heterogeneous response. And you may remember that's the same as what we found for the genotype. So we actually found the phenotype. So we actually found really good correlation between the genotypic response and the phenotypic response for PIDX1. The rank order for MSX2A actually is a little bit different than what we found for the phenotype. And that's likely because this trait is much more polygenic than pelvic girdle. So we were able to make this connection that there's strong divergent selection on MSX2A, as well as the underlying phenotype first dorsal spine. So given that we found this strong phenotypic and genotypic response, we decided to look at other traits that could be affected by predation, and I don't have enough time to get into the details of all of this, but we're able to detect direct and indirect effects on a variety of different things, including trophic morphology, differences in diet, brain size, pigmentation, and behavior between the trout and the control treatment ponds. And we were also able to detect some ecological effects, differences in turbidity, and as well as phytoplankton and zooplankton community. So in summary, we found that differential predation resulted in divergent evolution of first dorsal spine and its underlying gene, MSX2A. We found accompanying shifts in diet, trophic morphology, pigmentation, community structure, and turbidity. And this implies that predation has a variety of different effects on fitness that could be mediated through a broad suite of traits. So it could be the case that differential predation is really important factor in promoting divergence in the wild. And so it's not just resource competition that explains all of the fantastic diversification that we see. So for my postdoctoral work, I moved to a different question. I became interested in patterns of repeatability and asking why particular genes were more often used, and I'm curious about what genetic factors could influence patterns of repeatability. And this has been a fascination for a long period of time. I think a lot of attention was drawn to it after the Wonderful Life book came out by Stephen Jay Gould, because he posed this really interesting thought experiment asking if we were to replay the tape of life, would we see the same forms evolve? And a classic answer to that question has been no, but more recently there's starting to be a lot of evidence to suggest that's not the case, and we are seeing the same genes and the same phenotypes evolving repeatedly in a lot of different systems. And so there may be predictability. And predictability also has a practical application. So when asking or facing problems such as antibiotic resistance, climate change, and agricultural breeding, it could be really useful if we're able to say how deterministic is adaptation in the face of these challenges, and what factors are important to that predictability. So I, again, turn to Stickleback to address this question. And I, again, turn to the benthic limnetic pairs, because as I mentioned earlier, they're a really fantastic example of parallel evolution. And previous work had shown that up to 76% of morphological traits surveyed diverged in parallel for these pairs. And 49% of these parallel traits had a parallel genetic basis. So that really indicates that it's likely there's some predictability in the pattern of evolution. But what genetic and ecological factors determine this repeatability? So when I was considering different genetic factors to investigate, one of the things that I became interested in was pleiotropy, and the idea that it could influence a bias on which traits or genes are used over the course of adaptation. So in 1930, Fisher actually predicted that there would be a relationship between pleiotropy and the frequency of use during adaptation, when he had this relationship between mutational size and the probability a mutation is favorable. And he predicts this negative relationship where as mutational size increased, the probability a mutation would be favorable decreased. And what he meant by mutational size was that it depended on the number of characters affected. And that's most commonly called pleiotropy. So when a single gene can influence multiple traits. And empirical work is also suggesting that pleiotropy could affect adaptation. So it's been shown in a couple different systems that the evolutionary response observed depends on the amount of pleiotropy that a gene exhibits. So when there's high levels of pleiotropy, there's a low or small evolutionary response observed. So the genes that tend to be responding tend to have low levels of pleiotropy. So I wanted to determine whether or not this then lent itself to predictability. Was it the case that genes that have low levels of pleiotropy are more predictable? So the aims of the project were to quantify the repeatability of genetic evolution and then determine whether or not pleiotropy explains this repeatability. So I had samples from these 3 benthic and limnetic species pairs, and I had genotype data for 20 individuals. So I was able to estimate FST across the genome. And then I was able to quantify genetic parallelism, which I'll explain in a moment. And for this study, I also included stream-lake comparisons. So stream-lake ecotype pairs of stickleback have evolved many, many times across the world. And I had access to data from 16 watersheds, 16 of these stream-lake pairs. And again, I had genotype data from multiple individuals, so I was able to estimate FST across the genome. And what I did was that, so for each comparison, so we have benthic limnetic comparison 1, 2, 3, I marked a single nucleotide polymorphism in the genome as present or absent. So we have many different variable regions in the genome. I then used FST to determine whether the differentiation between the benthics and the limnetics was more than expected by chance. So what we generally call an outlier SNP. So something really extremely differentiated. And then I marked that in each comparison. And then I asked which of these outlier, so putatively adaptive regions occur multiple times across comparisons. And anytime I saw these multiply diverged loci, I marked them as parallel. It was a little bit more complicated than this, but this is kind of the straightforward, simple example. So jumping into the results for the benthics and limnetics, we have my parallelism metric on the y-axis and the different chromosomes. And what this is indicating is that these are outlier regions of the genome that diverge across multiple pairs. So this is where all of the pairs had an outlier region. So I was able to determine 170 regions of the genome that were evolving in parallel in the benthics and limnetics. And in the stream-lake data, there were many more pairs. So it wasn't the case that we ever had all 16 pairs diverging at the same position. However, many of the regions were repeated. So we found 377 regions of the genome that were evolving in parallel across stream-lake pairs. And pleiotropy is really difficult to estimate empirically. In an ideal world, you would knock out each gene one at a time and then estimate how many traits were affected. However, that's not feasible in an organism like stickleback. So I used a couple proxies. One was gene connectivity, so how many other genes does a single gene affect in a network? And the second was trait number, so how many traits map to a given gene or genomic region based on QTL mapping data. And the gene connectivity, I built a gene co-expression network to estimate. So my prediction is that loci with reduced pleiotropy will be more parallel. So just showing you the distributions of pleiotropy here, it's the case that most QTL or regions of the genome affect very few traits, so they're not very pleiotropic, but however, we have a lot of variants and there are some genes or regions in the genome that are highly pleiotropic. We see this for gene connectivity too, so most genes have very low levels of connectivity, but we have some really highly connected genes. So this was important for the study. Now jumping into the results for the benthics and limnetics, when we look at QTL trait number, we find we have these non-parallel regions and the parallel regions. And the parallel regions actually tend to, on average, contain loci that influence a greater number of traits. So this is opposite to our prediction. We find that parallel regions are more pleiotropic than the non-parallel regions. And when we look at the stream-like comparison, we find the same thing. So parallel regions of the genome influence a greater number of traits on average than their non-parallel counterparts. When we looked at gene connectivity, we found the same pattern. So the parallel regions on average tended to be more highly connected than the non-parallel regions in both comparisons, although it was only statistically significant in the stream-like comparison. And it's not the case that there's more genes in the parallel regions than the non-parallel regions. The gene density is completely the same between these two types of gene regions in both comparisons. So to sum up, there's considerable parallelism across the genomes of independent benthicomimetic and stream-like ecotype pairs, and these parallel regions tend to harbor genes influencing more traits and being more highly connected. So gene density doesn't explain the pattern. So we think it is the case that perhaps it is that these genes influence a greater number of traits. So the implication of this is that genomic divergence has a fair amount of repeatability and that there may be an optimal amount of pleiotropy. So beyond a point, it could be that pleiotropy may be a constraint, but low levels of pleiotropy may be actually advantageous. And you could imagine perhaps it's the case that being able to just adaptively change one or two or three traits at the same time could be really useful when you're trying to adapt to a new environment really rapidly. And I considered a bunch of other genetic factors that I don't have time to get into, but if you're interested in that, then I'm happy to answer any questions about that. So now I just need to acknowledge my PhD advisor, Dolph Schluter, who lab I was in for the first part of the talk when I conducted the experiment. And he was a really great PhD advisor, so I'm super thankful to him. Also to my collaborator on the project, Seth Redman, and all of the funding that I had during that time for this experiment. And part B, during my postdoctoral work, I was in Katie Peichel's lab at the University of Bern, and that project was funded by a Marie Curie action from the ERC, so I'm super thankful to them. And then thank you again to the Canadian Society for Ecology and Evolution. It's definitely my home society. It was the first scientific meeting I ever went to, so I'm really honored to be given this award by them. And thank you all for listening today.
Thank you, Diane. That was an amazing talk. Thank you so much. And I love the little shout out to CSEE at the end. That was, I'm happy that, I'm really happy you got that award, this award. Really awesome.
Yeah, I'm super excited that the next meeting is in Vancouver, so give me a chance to come back and see all my friends and collaborators there.
Yeah, definitely. Yeah. Thanks, everyone. Just as a reminder to everyone who's watching, if you have a question for Diana, you can type it into the chat and I will relay it. And if you have applause as well or clapping, then also post that. Always appreciated. There is some applause already filtering in. And one question I'll start off with. Given that only a fraction of important traits have been mapped in any population pair, do you expect the absolute level of pleiotropy to be much higher than the level currently inferred?
Yeah, so it's definitely the case that we're underestimating the amount of pleiotropy. And I think that might be hinted at by the difference in the distributions between the connectivity and the QTL. So the QTL distribution is pretty patchy with, it's just not a smooth distribution. So I think it is for sure the case that we're underestimating the amount of pleiotropy. But my inkling is, since we have this data from multiple comparisons and we see the same patterns with the two different estimates, that the pattern will hold even if we bolster our QTL data eventually.
Okay. Thank you. Another question on also related to connectivity. Connectivity dealt with gene expression. Is there any info on physical connectivity? That is, do parallel genes and proteins physically interact at all?
Yeah, so to my knowledge, we don't have that data for stickleback, but I'd be really keen to analyze that data if anyone knows that it exists, or in fact, in another system where I'd be able to do the same kind of analysis. That would be really cool. I believe that it's the case where there's data in similar systems in yeast, and where the gene expression data does correlate to physical interactions. And the reason I chose gene connectivity as one of my metrics was that there's data in yeast and in mice to suggest that pleiotropy is related to gene co-expression networks. So there is a relationship there, but physical data would be really cool to incorporate.
Another question coming in. What exciting new studies are you planning next? So
right now I'm definitely in this phase of going with the flow due to COVID and lots of other factors. So a lot of my plans for this year have been completely derailed. But one thing I'm really excited about is I have a really great postdoc who's really interested in gut microbiomes, and myself and one of my collaborators, Seth Redman, were interested in that during our PhD, but it was really just like a tangent. But we've thought of some really exciting field and lab experiments to do to look at estimating the consequences of different gut microbiomes in stickleback. So we want fitness estimates, and we want to look at ecological consequences of gut microbiome differences. So we're really excited about doing that.
Yeah, wow, that sounds really neat. I'm excited to hear more about it in the future. Maybe when we have a mid-career award, you'll be able to give a talk about that. You mentioned for the second part that you looked at a bunch of other different genetic factors. I was just a little curious about like what, if you could briefly like mention what they were.
Yeah, so one of the things was recombination rate. So we had data from another study showing that recombination rate really influences which parts of the genome are diverging, especially when there's gene flow. And so we thought that that might play a role in predictability, so whether or not you repeatedly see divergence. And we found that it was the case for the benthic limnetic pairs that it played a role, but for the stream-like pairs, it didn't. And we're trying to understand why that relationship differs between these different geographical contexts. And then we also looked at mutation rate, and we found that that didn't differ between the parallel and non-parallel regions of the genome, whereas theory would predict that it could play a role. So that's one of the cool things.
Neat, yeah. I'm curious as well. Just if you is stickleback your ideal model organism, or if you had another one you could work with, what would it be?
So yeah, I've worked with stickleback for 10-ish years now. And so they're very near and dear to my heart. And I think I'll always work on stickleback, at least to some extent. But I have a new grant and a collaboration with one of my colleagues at UCSD where we're working on chubs. So I'm excited to go out and find some chubs and do some genome sequencing of them, because I haven't looked at any other fish taxa in a long time.
Awesome. Another question in, would you expect loci with lower levels of pleiotropy to exhibit more parallelism after more generations pass?
Yeah, that's an interesting question. I don't think I've thought about that for long enough to give you an off the cuff answer to that. My biggest question about the pleiotropy pattern is whether or not it will hold in other systems, because my inkling is that in stickleback, this pattern might be at least partially due to standing genetic variation. So it might be the case that over a long period of time, recurrent episodes of selection and migration in stickleback has allowed it such that you can have these loci that have higher levels of pleiotropy where it's synergistic. So they're working together instead of traits being favored in different optimums, which is when the theory suggests that it will become constraining. So I'm very curious to see if standing genetic variation is one of the key factors in stickleback. So, and I think that could also play into these timescale questions. So I think looking at in other systems might help to inform that question, but I would be keen to look across different stickleback systems where there's different timelines and see how much variation there is, but I don't think I have a good answer for that right now.
That was good. Nice off the cuff one. Yeah. In the pond experiment, did you see any patterns with lateral plate variation between treatments?
So I've only really briefly looked at the lateral plate data actually, and I have my master's student in the lab is actually doing that. So when I looked at plate number, I didn't find any strong effects. It was like pelvic girdle. So in some of the ponds, I think it was like 3 of the ponds, it was favored of more lateral plates with the trout, and one pond, it was in the other direction. But one of the things I'm interested in is whether or not lateral plate number and plate area give different signatures, because it's the case that you can have an individual that has fewer plates cover a larger area. So there's residual variance between plate area and plate number. And so I have what my master's student is looking at that. So hopefully we'll have an interesting answer for you soon.
Sounds good. Yeah. I was also curious, you mentioned that you also look at in the first part, not just predators, but also parasites and kind of other interactions as well. Would you be able to briefly like mention that as well?
So that work's all been done in collaboration with Dan Volnick. So Dan's lab is really interested in this parasite, Schistocephalus, which is this internal parasite. It's this nasty little worm. And if you want to be grossed out, you should check out some of his videos where he cuts open fish and there's all these things coming out of them. It's like some sort of alien invasion thing, really disgusting. And so I did a lot of genetic work for him, doing genome-wide association mapping, trying to look at whether or not there's a parallel genetic basis for resistance to those parasites. So some populations are just loaded with these worms and other populations have no worms. And so well, Dan mostly, and just me helping him, is trying to understand why there's this pattern of variation, what the genetic basis of that resistance is.
Okay. Yeah,
and
really.
we found some candidate genes, so that's fun.
Oh, wow. Okay. Awesome. We'll give you one or two more questions. One in, are the experimental ponds still in use?
Yeah, so Dolph replaced me and all of my colleagues, of course, when we left, and there's a great new cohort of students that are conducting pond experiments, and one of them is Ken Thompson, who was the doctoral prize winner this year. So Ken's doing some really cool stuff there. The ponds are amazing. I mean, I love Dolph, but I definitely wanted to work in the ponds as well. So it was 50% loving Dolph, 50% wanting to do an experiment in the ponds. And so Dolph's still doing some really exciting stuff there as well.
Thank you for your honesty as well. And as a little plug as well, if you're interested in these ponds more and you want to see Ken's talk and you haven't seen it, then you can check it out on our YouTube page as well. Awesome. I think we'll let you go now. Thank you for answering all of these questions. Thank you for an amazing talk. You really set the bar high for this Early Career Award series.
I'm super stoked for Kyoko. Kyoko and I have known each other for many, many years, and so I'm super stoked to hear her talk next week, or next week, or the next week, soon.
Next week, yeah. Next week, July 3rd at 4 p.m. same time. We'll have another terrific Early Career Award talk. So please tune in again. And on that note, thank you everybody for watching.